everyone and welcome back. This video contains the hot topics in immunology, so it is not for the freshers. If you want to revise immunology, you can join. Let's start. Here we go. MHC pathway specialization. There are three types of MHC classes: MHC class 1, 2 and 3. Here MHC class 1, it's found on almost all nucleated cells and it presents endogenous antigens, that is intracellular antigens, to CD8 plus bearing T cells, that is the TC cells. So MHC class 1 presents endogenous antigens to CD8 plus bearing T cells, that is the TC cells, and its structure include an alpha chain plus a beta 2 microglobulin. So this is the main points related to MHC class 1. Now let's check MHC class 2. It's found only on professional antigen presenting cells, APCs. So, MHC class 2 is found only in antigen presenting cells like dendritic cells, B cells and macrophages. And it presents exogenous antigens. The difference is MHC class 1 presents endogenous antigens, but class 2 presents exogenous, that is the extracellular antigens to CD4 plus T cells. CD4 bearing T cells, that is the TH cells. So MHC class 2 presents exogenous antigens to CD4 plus T cells, that is the TH cells, and its structure include an alpha chain plus a beta chain. Okay, class 1 is present in almost all nucleated cells, present endogenous antigens to CD8 plus T cells, that is the TC cell, and class 2 it's found only on professional antigen presenting cells and it presents exogenous antigens to CD4 plus TC cells. Okay, that is the main points in MHC. Next, hypersensitivity. Hypersensitivity. Let's check. There are 4 types of hypersensitivity, that is type 1, type 2, type 3 and type 4. 1 hypersensitivity is immediate and that is IgE mediated. Type 1 is immediate and IgE mediated, and example is anaphylaxis and asthma. Anaphylaxis and asthma is an example for type 1 hypersensitivity which is immediate and IgE mediated. Then type 2. Type 2 is cytotoxic. Type 2 is cytotoxic which is antibody mediated. Type 2 is antibody mediated. Example is transfusion reactions, that is blood transfusion reactions, erythroblastosis fetalis, etc. So type 1 immediate IgE mediated, example anaphylaxis, asthma. Type 2 cytotoxic antibody mediated, example blood transfusion reactions, erythroblastosis fetalis. Type 3 immunocomplex mediated. Type 3 immunocomplex mediated. An example is arthritis reaction, systemic lupus erythematosus. Systemic lupus erythematosus. Then type 4. The type 4 is delayed type hypersensitivity. Type 4 is delayed type and it is cell mediated or T cell mediated. An example is contact dermatitis. Contact dermatitis, that is contact dermatitis and tuberculin test. Example for type 4 is contact dermatitis and a tuberculin test. Okay, so there are 4 types of hypersensitivity, that is type 1, 2, 3 and 4. Type 1 is immediate, type 2 cytotoxic, type 3 immunocomplex mediated, type 4 delayed type. And type 1 is IgE mediated, type 2 antibody mediated, type 3 like immunocomplex mediated, type 4 is cell mediated. The antibodies involved in these hypersensitivities are type 1 IgE, type 1 IgE mediated, type 2 IgG and IgM mediated, type 2 IgG and IgM mediated. Next, transplantation. The first human kidney transplantation was done by Joseph Murray. He transplanted kidney between identical twins. So here are some transplants. Let's check the difference between the transplants. First, autograft. Autograft are self-to-self transfer. That is, autograft is self-tissue transferred from one body side to another in same individual. Example, transferring healthy skin into burned area. That is an autograft. Okay. Then isograft. Isograft is between genetically identical individuals. That is syngenic. That is the transplantation between twins. Graft between genetically identical individual. That is graft between identical twins is isograft. Next, allograft. Allograft is between genetically different members of the same species. For example, skin transplant between two individuals of different genotype. Then xenograft is between different species. Xenograph graft is between different species. Okay, once more, autograft self to self, isograft between genetically identical individual, that is identical twins, allograft between genetically different members of the same species, then xenograft between different species. Next, complement system. There are three types of complement pathways. These are classical pathway, lectin pathway, and alternative pathway. The classical pathway is antibody-dependent pathway and it is triggered by the formation of soluble antigen-antibody complexes. So, classical pathway is triggered by antigen-antibody complexes and is involved by IgG and IgM. Classical pathway involves IgG and IgM antibody and it is triggered by antigen-antibody complexes. Note the point, antigen-antibody complexes is triggering the classical pathway in complement system. Okay, so the antibody involved in classical system, classical pathway is IgG and IgM. Okay, then lectin pathway. Lectin pathway. It is antibody independent pathway. Lectin pathway is antibody independent pathway and it is triggered by MBL binding to mannose on microbial surfaces. Lectin pathway is triggered by MBL binding to mannose on microbial surfaces. So, what is MBL? What is MBL? Mannose binding lectin. MBL is mannose binding lectin seen in our blood. So, MBL is seen in blood. Form is mannose binding lectin. So, this mannose binding lectin binds to mannose on microbial surfaces. MBL binds to mannose on microbial surfaces and this triggers this lectin pathway. Lectin pathway is triggered by MBL binding to mannose on microbial surfaces. Okay. In the three pathways, that is the classical and the lectin pathway, in classical and lectin pathway, the C3 convertase is formed of, C4b plus C2a. C4b plus C2a composes the C3 convertase. Note the point, in classical and lectin pathway, the C3 convertase is formed of C2a and C4b. Okay, next, alternative pathway. The alternative pathway is triggered by spontaneous C3 hydrolysis. Alternative pathway is triggered by spontaneous C3 hydrolysis on foreign surfaces. C3 hydrolysis is called tick over. Here the C3 is always active and it hydrolysis on foreign surfaces and this triggers the alternative pathway. In alternative pathway, the C3 is stabilized by properdin. In alternative pathway, C3 is stabilized by properdin. So, these are the main points in complement system. The common result is the formation of C3 convertase and eventually the membrane attack complex is formed. The membrane attack complex, MAC, membrane attack complex, it is composed of C5b, C6, C7, C8 and C9. Membrane attack complex composed of C5b, C6, C7, C8 and C9. Okay, these are the main points in complement system activation. Here is some vocabulary list related to immunology and the first one is hapten. Haptens are a small molecule that is antigenic but only immunogenic when attached to a large carrier. So, haptens are small molecules that is antigenic but only immunogenic when attached to a large carrier. Next, adjuvants. Adjuvant is a substance added to a vaccine to enhance the body's immune response to an antigen. Adjuvants are substances added to a vaccine to enhance the body's immune response. So, these are substances added to vaccine to enhance the body's immune response to an antigen. An example for adjuvants is alum, aluminum potassium sulfate. Aluminum potassium sulfate, alum is an example for adjuvant. Then secretory component. Secretory component. This is a polypeptide that wrap around dimeric IgA. Secretory components are seen in IgA. These are polypeptides that wrap around dimeric IgA to protect it from digestion by enzymes in the gut or respiratory tract. So, secretory components is seen in IgA. It's seen wrapped around dimeric IgA to protect it from digestion by enzymes in the gut or respiratory tract. Next, anamnestic response. Anamnestic response. It is the rapid and intense secondary immune response following re-exposure to an antigen. Anamnestic response is the rapid and intense secondary immune response following re-exposure to an antigen. Then hybridoma. Hybridoma is a hybrid cell used to produce monoclonal antibodies. Hybridomas are hybrid cells used to produce monoclonal antibodies formed by fusing a specific B cell with a cancerous myeloma cell. So, hybridoma cells are formed by fusion with a B cell with a cancerous myeloma cells. Next, Hat medium. Hat medium is a selection medium used to isolate fused hybridoma cells. Hat medium is a selection medium used to isolate fused hybridoma cells, and it is composed of hypoxanthine, aminopterin, thymidine. Hat medium composed of hypoxanthine, aminopterin, thymidine. Then opsonization. Opsonization is the process of coating a pathogen with the antibodies. Coating a pathogen with the antibodies or complement, that is the C3b. The process of coating a pathogen with the antibodies or complement to facilitate phagocytosis. So, opsonization is the process of coating a pathogen with the antibodies or complement to facilitate phagocytosis. Next, energy. Energy is a state of immune unresponsiveness. Energy is a state of immune unresponsiveness where a T cell failed to respond. Where a T cell failed to respond to its specific antigen due to lack of co-stimulation. So, energy is a state of immune unresponsiveness where a T cell fails to respond to its specific antigen due to lack of co-stimulation. That's all for today's class. If you want to attend more questions in immunology, you can join my Telegram channel. If you are preparing for Neet, JEE, CSE or NET in life sciences, here's the best option for your preparation, the Akans Life Science Academy. You can call in the given number for more details. Thank you.